Fermentation has been around for, Christoph, correct me, but I would say thousands of years by now. So you might expect that we know a lot about it, but in fact, nowadays, researchers are exploring how microbial communities can be intentionally used to design and improve food characteristics and also potential health outcomes. So it's my pleasure to introduce Christophe Cotin. He is our first speaker and professor at the Laboratory of Food Chemistry and Biochemistry at the KU Leuven in Belgium. His research focuses on cereal compositions and dietary fiber, the enzymes that degrade them, and microorganisms in cereal processing and human health. And Christoph is also the coordinator of the EU-funded project Health Firm, which is working on plant-based food fermentation and health. So, Christoph, I'll hand over to you, and please feel free to start.
Thank you, Stefan. I hope in the meanwhile you can see my screen. It should be shared. Yeah, perfect. So good afternoon, everybody, to this webinar also from my part, and I'll be happy to explain some of the things we are doing, not per se in Health Firm, but as a whole with regard to traditional fermentations. And so the title of my talk is going from spontaneous to rationally designed plant-based food fermentations to boost product quality and health. And if you break it down, you could ask yourself, okay, why plant-based? I think most people on the call might already be convinced that we need to transition in part from a more animal-based diet towards a more plant-based diet for sustainability reasons, for the health of our planet, but also for our own health. And that is very nicely written down by the Eat Lancet Commission in one of their also latest papers in the Lancet in 2025. And one of the conclusions they come to is that today we eat, as I said, too much meat, and we have to increase our consumption of whole grain products and also of legumes. And maybe something that a lot of you don't know, but these are already staple foods for the majority of the world. And if you look at grains, for example, we produce approximately 1 kilogram of grain per person per day. So it's clear that based on this, they can be really the cornerstone also going forward for a healthy and sustainable diet. And so the focus that we have is on those grains. So on the one hand, cereals, on the one hand, legumes. And I think the table that I show you here, where you have whole grain intake estimated or advised by the Eat Lancet Commission, that it clearly shows that at least 1/3 of our diet should be whole grain, a good portion should be legume-based. That being said, grain-based foods, quite nice. We eat them already a lot, but if we go into newer products like yogurts, like oat-based drinks or pulse-based drinks, there are still some issues. And one of those issues is off flavors. Another issue is that if we try to extract components from those materials, like proteins, like fiber, extractability is very often quite low. We also have a lack of, for example, vitamin B. Mineral bioavailability is low. So there are challenges that we need to meet. On the other hand, there's also a lot of opportunities. You might realize it or not, but we do have a rather large fiber gap in most of our diets, which means that instead of consuming between 25 and 38 grams of fiber per day, in a lot of countries, we only reach 15 to 18 grams per day. So changing from more animal-based to more plant-based, we can have more dietary fibers in our diets, and that is a very big benefit also. So challenges, opportunities, and we believe that to tackle some of those challenges and make use of those opportunities, that fermentation can give us a big hand. And if we talk about fermentation today, the word is a bit loaded because it's actually covering a lot of different things and quite different things. So when I talk about food fermentation, I talk about the traditional fermentations that Stefan properly mentioned, like being already millennia amongst us. I'm not talking about biomass fermentation, where you create, for example, proteinaceous biomass by cultivating cells. And I'm also not talking about precision fermentation, where fermentation by a microorganism is used to produce very specific components that are then harvested and marketed as such. So I'm talking about traditional fermentation, which is defined by ISAB as foods that are made through desired microbial growth and enzymatic conversions of food components. So we use microorganisms to change the matrix of the food that we are making to maybe make proteins more soluble or to make it more acidic, more safe. So that type of fermentations, that is what we're looking into. Why do we think this is of interest? Because again, I think there's a lot of opportunities, but also the majority of people feels very strongly about fermentation. They think it is good for health. They also believe that it's good for taste. So there's a real, I would say, hype almost going on about fermentation, with a lot of people buying into the knowledge there is and buying into possible benefits of those fermented products. Now, that being said, there's still a lot that we need to learn. On the one hand, we have been doing fermentations for millennia, but that has always been very spontaneous fermentation. So microorganisms that are present on the raw material, we let them do their thing, enrich themselves, and ferment products. But very often we don't look at the matrix. We let those microorganisms do their trick, but there's sometimes insufficient knowledge on what is there from a food technological point of view or food chemistry point of view. So those biochemical conversions that are happening, I think we need to study more. But maybe more important for the topic of today is that if you want to do microbial fermentations, maybe we can do much more than what we do today by relying on spontaneous fermentations and the microorganisms that are already present on the raw material. There's a huge diversity of microorganisms out there, and we know only a very, very small part of it. And I'm very sure that there's a lot of microorganisms out there that might do a much better job than the traditional spontaneous microorganisms that we find. And so exploiting the microbiome diversity for food fermentation is, I think, something that a lot of people are looking into, including ourselves within the Health Firm project and sister projects like Domino and Wheat Biome and so on. Now, if we have microorganisms that are of interest, well, there's maybe even more that we can do, which is instead of relying on spontaneous combinations, also try to design consortia and say, okay, we are going to use one microorganisms for specifically this purpose. We're going to use another one for another purpose. And so we combine microorganisms to get to designed consortia to then do fermentations that are optimal with regard to either product quality, taste, sensory, a nice buttery flavor or nice other flavors, or with regard to health, so that we are producing, for example, vitamin B12, or that we are solubilizing fibers so that they get more active in our gut, feeding into the gut microbiome and so on. And so that is the belief that we have, that we need more types of microorganisms, but that we have to also combine these in defined consortia to do a better job at fermentation and to bring us more health benefits than what we have today. So that is what we try to do. And I'm not going to talk about Health Firm as a project as such much more than this slide. So what we are doing in the project is looking at those microbial resources. Can we find new microorganisms? We are looking at fermentation technology. How can we put those microorganisms to proper use to produce better products, make sure that people appreciated legume-based products more than sometimes they do today by altering flavor profiles, taking away some off flavors and so on. We also look through a lot of intervention studies on humans into human health and the gut microbiome, and we also look at consumer behavior. So these are 4 elements, and I will tackle 2 or 3 of them now. Because how we try to come up with diversity is by asking, or we did that by asking people all over Europe to send us samples of their fermented foods. Because there's a large diversity, we want to capture part of that. So we asked people around Europe to send us products, and then we took apart those fermented products, looking at all the different microorganisms that are present. And so for that citizen science project, yeah, which was a lot of work to set up, I can assure you, but which also gave very nice input from consumers, small companies all over Europe. And so we were able to gather around, let's say, a small 700 sourdough samples, additional 200, 300 other fermented samples. And you can see here on the graph where all those materials came from. So from all over Europe, some even from other parts of the world. And so these samples were taken apart by several labs in Europe, looking at all of the different microorganisms in there. And I can assure you that's a very rich source of material. And so what we have been looking for is, can we find genetics or genes in those microorganisms that will produce additional dietary fibers, exopolysaccharides? Do we find genes that allow short-chain fatty acid production? And I'll come back to that a little bit later. Vitamin B production, carbohydrate utilization. So through in that whole set of thousands of strains, we looked for which strains can help us based on their genetics, but also based on other type of experiments. Phytate degradation for mineral bioavailability. So all these elements were looked into. And so that means that we identified a lot of microorganisms that could be of potential use. And we checked them for that use, but we also checked whether we could indeed make those rationally designed consortia that would do an optimal trick in fermenting into a good product, but at the same time possibly making an even healthier product. And so that I can assure you that takes a lot of experimental effort. And here you see results from the University of Bolzano, for example, where they combined up to 6 different microorganisms with different properties and combined that with different matrices, be it completely whole meal flour, whole meal combined with faba bean flour and concentrates to see which fermentations went well, and also have a look at the health outcomes. And so protein digestibility, we could see was affected by a specific consortium, not by others. Phytic acid degradation was affected, so possibly increasing mineral bioavailability, which is a very good thing if you look at plant-based foods, because most of the minerals are sequestered with phytate, and so not very easy to absorb in our body. And so fructan content, for example, that for people with IBS could lead to pain and bloating. So a lot of interesting results were found there. And so we can use those newly identified microorganisms and combine them into rationally designed microbiomes to do that traditional fermentation, leading to better outcomes than what we have today. And the second example is here, that was not for sourdough, but here in this case for cereal pulse-based beverages. Like I told you before, a lot of the beverages, plant-based beverages that we have today on the market are relatively low in fibers, in proteins, in nutritious components. And so we wanted to see whether by using the same approach as I showed you on the previous slide, we could come to higher quality products from a nutrition point of view, from a health point of view. And so also there, we looked into binary cultures, combining 2 microorganisms, combining 3 microorganisms, combining 4 microorganisms to see whether we could have indeed benefits. And also here, we were able to show that by using a rational design of that microbiome that we're going to use, that we are able to get to the results that we want to. And what is one of those results? And then I come to the last 2 slides of my presentation. That is, for example, short-chain fatty acid production. So we all know that if we ingest fibers and they come into our gut, part of those fibers can be fermented into short-chain fatty acids, which are good for our system. The question is, if we produce these short-chain fatty acids already in our product, in our fermented food, how are they going to affect the health status of our body? And so these are trials we did where we measured the impact of short-chain fatty acid intake versus short-chain fatty acid production in the colon on elements like GLP-1, which you all recognize as glucagon-like peptide 1, so the molecule that Ozempic and Wegovy and others are based on. And what you see here is that the ingestion of those short-chain fatty acids indeed leads to increases of some of those satiety hormones, which is leading to an effect that is to a much smaller extent, but similar to what an Ozempic is doing in our body. And so what we see here is that through targeted fermentations, we could achieve products with, for example, increased short-chain fatty acids that could lead to increased production of this type of satiety hormones. And then the question is, of course, does it really affect a human body? And here you can see some of the subjective appetite scores that were recorded with the products that we just saw, those capsules. And then you see, for example, in the upper left corner, that the hunger feeling, if we have short-chain fatty acids being taken up in the small intestine, are significantly reduced compared to a placebo, so leading to less hunger and therefore typically less food intake. And that we also see that on prospective food consumption, that also there you see a difference, so that people have the intention of eating less after having those short-chain fatty acids taken up in their small intestine. And so I hope that with the very broad strokes that I told you about today, that I've given you some insight in how we try to transition from, let's say, traditional spontaneous fermentation into still traditional but designed fermentations by making full use of microorganisms, combining them into rationally designed consortia microbiomes to do fermentations for us that then can lead to health benefits. So I hope with this that I gave you some picture of how we try to do this and of some of the results. And if there are questions, I'm of course very much open to answer them at this moment. I will unfortunately not be able to join the panel later on, but if you have questions, I'm more than happy to answer them now.
Yeah, thank you very much, Christoph. That was great to hear. I always find it super fascinating when you talk about the project that you capture this somewhat fleeting knowledge that you have that we have out there with all the citizens and how many citizens you integrated in such large scale in your project. So very exciting. Looking at Deborah, do we have questions for Christoph from the Q&A or from the registration maybe even as well?
No, we don't have questions from the Q&A, but I have a question that was sent with the registration. Is what is currently the biggest barrier to bringing next generation fermented foods to market? I know it's quite broad. If you could try to.
That's a difficult question. So I think there's already starter cultures, of course. So we have spontaneous fermented foods. We have starter culture initiated, but there very often it's one microorganism that is used. So if we want to go to more microorganisms combined, I don't think there's a problem or a barrier to doing that legally if we combine microorganisms that are known. If we go into new microorganisms or microorganisms that have been genetically modified, then of course the applicable legislation would apply. So that is not something we're doing in HealthFirm. But for the rest, I think there's a lot of good opportunities for next generation products to come on the market. One issue is that we're not always allowed to talk about it. From a legal point of view. I'll just give you one example. We can talk about fiber content of a product. If we change that fiber from insoluble fiber to soluble fiber, which is more prebiotic, we cannot talk about the fact that it's more prebiotic because EFSA does not allow it. And so I think that is a little bit of a barrier. We go into what we believe are more nutritious products, if we believe that there is sufficient evidence. But if the government does not allow that evidence or does not allow the claims yet, then it's more difficult to communicate about it. And that is to some extent a hurdle, I think.
Thank you. It was difficult, but your answer was clear.
Yeah, I agree. I think the answer was spot on. Thank you so much for this insight. So I would suggest that we then move on to the next speaker. But in case you have more questions for Christoph, Christoph, if I understand you correctly, you will still stay for a little bit. You can have a look at the Q&A section as well and maybe answer those questions that are addressed to you. And in the meanwhile, we can then move on to our next speaker and to move further upstream to the food system, basically back to where I promised we would start today, to the agricultural environment, where microbiomes also can help shape crop productivity and resilience. And today we basically have two case studies from major crops with us. The first one being soybeans, a key crop for food and feed production worldwide, and with also increasing demand. And I'm sure I'm not the only one who have noticed the empty tofu shelves in the supermarkets lately. But looking at how microbiomes can impact that, if we could understand how these microbial communities support the plant's nutrient cycles, plant health, and resilience towards external factors, that would already bring a great contribution to more sustainable and resilient agricultural systems. And to tell us more, I'm pleased to welcome Anastasia Juskovic. She's a PhD student at the Austrian Institute of Technology in the Department of Health and Bioresources, and she is working in the EU project Microbiomes for Soy. Anastasia's current research is focusing on plant microbiomes and specifically the interactions between soybean plants and their associated microbial communities. So Anastasia, please feel free to start your presentation.
Give me a second. Hope everyone sees everything well enough. Okay, first and foremost, thank you so much for the introduction. I am Anastasia Juskovic, and as you've heard, a second-year PhD student at the Austrian Institute of Technology. And the bulk of my work is done within the Microbiomes for Soy project. And today I will be happy to present the role of the microbiomes in sustainable soybean agriculture. Well, cultivation in general. But what is important for me to note from the get-go is that this story is quite old, especially with soybean, because it's a highly specific plant, as I will describe later on. But first, we have to describe our emerging threats, or what is currently happening, and why do we point a finger to sustainable agriculture for help. So first of all, we are currently in large-scale production dealing with large climate constraints, agricultural production that is oftentimes not sustainable, which leads to degradation of soil as well as soil fertility, which in turn increases the diseases in plants, loss of biodiversity, causing a global security problem, as my colleague previously also stated. We also have the fact that the diets are very unhealthy and unsustainable. All of this results in both the policy and societal pressure that should lead towards a search for new biological solution. To the credit to the many other scientists and agricultural workers that came before me, there are many things that are already being done about it, from growing different breeds of not only soybean, but all the other plants, to different forms of fertilizer. Many things are being done. In my case, we are looking at, I would say, somewhat new, one of the most researched aspects in several last decades in science, a new aspect which could help us tackle this issue, which are the microbiomes. So I will be focusing on microbiomes in plants today, but in general, microbiomes are a group of bacteria, fungi, viruses, so microorganisms in general that live at a certain place at a certain time and create large microbial networks that can, within themselves, they can have many different relations, whether it's commensalism or parasitism, or so on and so forth. What is important that these communities are highly established in almost every single part of the plant, from the soil to the root. Recently, one of the novel things was that up until recently, seeds were mostly considered sterile, or the seed microbiome was considered irrelevant, which is recently we're finding out that is certainly not the case. So this is a tool that we are choosing to look at one of the most important crops in the world, which is soybean. And the reason why we talk about it and why we study it so much, it is one of the largest produced crops in the world. It is extremely high in quality protein and contains all the essential amino acids for human needs, as well as animal, which leads me to so many diverse applications that this crop has, from deriving oil to biofuel, to extreme amounts of animal feed, and like more than half of soybean is used for animal feed, to increase demand in human consumption. All of this connected shows that we have a rising demand to produce soy just about everywhere. So soybean is a native plant in mostly East Asia. It was domesticated in China, and currently it's mostly produced in Brazil, Latin America, and USA. And what is important to note, and how we were able to spread this crop so worldwide, is the trait number 5, which is the fixation of atmospheric nitrogen, which is a trait that soybeans have alongside many other legumes. So as you know, nitrogen is in the air all around us, but in order for it to get fixed into more bioavailable forms like ammonia or nitrate, it needs the help of bacteria that this plant has co-evolved with. So rhizobia is a group of bacteria that can fulfill this function. And as I've said, they have co-evolved with soybeans to form small nodules in the roots, especially in later stages of growth, which allow this plant to be supplied with nitrogen. I don't know how acquainted you are with this, but soybeans as crops require an extreme amounts of nitrogen, way more than wheat, for example, also due to their very high protein content and many other factors. And the inoculations or the additions of microorganisms to soybean, as I've mentioned in the beginning, is a very old story. The first inoculation with these bacteria that were isolated as culprits to fix nitrogen started actually in the early 1900s. And so we see the application of microbiology in the fields before we even had microbiomes defined as a term in the first place. This is, I would say, a step 1 in implementation of sustainable agriculture. And as you can see, the difference between uninoculated and inoculated soybean is extremely high. Of course, this is currently being used to perhaps lower the use of nitrogen fertilizer, which whilst when they were invented the first time, they created a true agricultural revolution. However, they're extremely cost, energy, and as well as they emit a lot of greenhouse gases when they are created, so minimizing the use is generally good. And now I can say we move on to step 2. After a while, we use valorising, and then people ask what other bacteria could we utilize in this way. So this is a picture from the figure from the paper that was published on field trials in Brazil. And they're one of the primary researchers that currently deal with plant growth promoting bacteria or plant growth promoting rhizobia, PGPBs or PGPRs. So what was first tried is that instead of just using one highly effective Bradrhizobium, which is bacteria from the Rhizobia group, they co-inoculated with several of them and noticed that the results were very good. Bigger roots, general biomass, larger yield, the nitrogen present in the grain was also higher. And then several other bacteria were also accepted as plant growth promoting bacteria, like Azospirillum is very famous. I think currently 25% of all crops, all soybean crops in Brazil are co-inoculated with both Bradrhizobium and Azospirillum, as well as many other like Penibacillus is very good in potassium and iron solubilization, phosphate solubilization by Bacillus. So first time we are exploring the way that these bacteria function together and allow us to generally get so much better results. And this leads us to perhaps the most important part of this presentation, which is the bacterial consortia, as you've heard in the previous presentation as well. One of the most studied topics in microbiology currently. If you've read about them before, they were very often referred to as syncoms or synthetic communities of bacteria that were combined in a lab that could perhaps have an even better effect than single inoculates. Now, why do we want to move from single to multiple is generally a big question. So whenever we talk about inoculates or anything that's done within a field, we are talking about adding a new bacteria into an already existing system. So we have, as I've mentioned, the root microbiome, the soil microbiome, the microbiome of the plant, and the plant's own immune system, and so on and so forth. So in order to ensure that the inoculants are effective, they need to be applied at a certain time, in a certain place. Also, in some fields, they may be more effective, in some may be less. So when we make a consortium of bacteria that each fulfill a different role, it increases a chance that there will be more success and that the community would be more stable or so easily or modified. Like the figure that I'm currently presenting was in a paper where they did basically, they omitted essential fertilizing from plants, their own vitamins and minerals that are needed, and then they inoculated with different groups of bacteria to see if it would allow them to grow better or basically make up for the lack of nutrients that they were presented with in their soil. And it has proven to be generally quite highly effective. So what is important to note here is that there is vast evidence of plant beneficial effects in both lab and greenhouse trial when we talk about consortia. And they affect so many different traits like root architecture and plant biomass, yield, general phytoremediation and pathogen protection, and so on and so forth. However, it is important to note that the consortia studies are still very much ongoing, especially how to form them, which bacteria to use, how to apply them in a way that would be acceptable in large-scale field trials. And there is also some legislation issues in terms of like previously when we had single inoculant, it was very easy to commercialize it when with consortia, because again, we are talking about alive systems that go into other alive systems. There is horizontal transport of genes and many other things that happen that still need to be firmly defined before this is a commonly used thing, but certainly an extremely high potential one. So overall, I would like to also use this opportunity to present the project that I'm a part of. As you've heard, it's a Microbiomes for soy. And similarly to this webinar, the project itself deals with microbiomes from the primary production to the very end consumption. So we deal with soybean microbiome as well as the soil, to the way that soybeans can be fermented to make novel-based aquafeeds as well as food, and how this food influences the gut microbiome later on, hoping to move towards the so-called Food 2030 priorities, which is organic production, lowering food waste, and increasing biodiversity. My part of the project generally does deal with primary production, and I will not talk much about my results, but overall, we do deal with the observation of microbiomes in the way that they are influenced by agro-management practices, microbial inoculates, and the influence of soil environment on how soybean grow. And this is all based, this is a European Union funded project, and it's a European-based project, because as I've said, this is a very important crop, and to develop more independence in producing it locally is generally very, very important. So I would like to leave you with just a future outlook and what we could expect from basically microbiomes applied as a tool for sustainable soybean production. So most of all, we will pivot towards microbial consortia. There is a development of newer inoculation methods, which I think is very interesting. Previously, seeds can often be coated. The soil can be granulated. For example, that's a common practice in France. For example, in Austria, they do more of a seed coating. There is also foliar exposure. So people inoculate the leaves, and then afterwards that gets absorbed. And a very new thing, like for example, there's a very good company here called Ensemo, which does for example, small cuts in the seeds, which get inoculated with bacteria and then sealed, which prolongs the shelf life, which has, believe it or not, been one of the bigger issues with inoculation. Hopefully reducing fertilizer dependence, as well as stress resilience increased in plants. And also fifth, and one of also the most promising, microbiome-informed breeding that we would select for plants that would favor certain microbes and hopefully change the way we grow them, because informed breeding was a thing that we have done as people for a very long time. Hopefully, this will be a new piece of the puzzle that we could observe. I would like to just quickly thank my PhD supervisor, Angela Sesic, as well as Federica Zanetti and Miriam Avencino as my closest collaborators, and the entire Microbiomes for Soy team, as well as my colleagues at AIT. I would not be able to do any of them. And thank you for the organizers and you for your attention. If you have any questions, send them my way.
That's brilliant, Anastasia. Thank you so much. That was great. I think it's always so fascinating to see the complexity of these microbial consortia that you mentioned, and understanding what features into what and what impacts what. So that's really, really interesting work. I can imagine quite demanding, but also extremely fascinating. So thank you so much, Deborah. I think we have a couple of questions for Anastasia, right?
Yes, there is. The first one
Yes,
is,
yeah.
soybean is also very demanding in water. Is there a that could be useful and less water demanding?
Yes. So, as I said, a lot of these things are still ongoing. I think this was a Pseudomonas putida co-inoculation with Bradyrhizobium, which was named as a species that was able to mediate drought stress in soybeans, though this was a greenhouse trial from, I think, 2 or 3 years ago. So there is a lot of work being done with that. And I also know for a fact that there's a lot of soybean currently being grown to be much more sturdy and to generally give out less water via evaporation. There's a lot of projects dealing with like several, they're called like soybean in different decades, basically, into trying to make soybean much more resilient. But yeah, it's a very water demanding plant.
Okay, and do you have time for another question, Stefan? Okay, one question that was sent. What do you see as the main challenges in proving causal links between soil microbiomes, crop quality, and human health?
Crop quality and soil health, I think there is no challenges there. I think that's decently proven and so on and so forth. There are papers that analyze, for example, soil diversity connected to human health quite often. The issue is it's always, I mean, I hate to say it, but it's a multifactorial aspect. We can compare it, and I think there absolutely is correlation. However, we are battling with so many different things here, personal habits, diet choices, availability of healthcare in the region, and so on and so forth. So I think those are the major challenges, but there's a lot of work being done. So I don't think it's impossible.
One last question. But does the co-inoculation affect the nutritional properties of soybean?
Sometimes yes, actually, in a very good way. When they inoculated, I think it was with the Azospirillum brasilense and Bradyrhizobium japonicum, the grain had more nitrogen in it, and I think the general protein content of the crop has increased. Though none of these are, you know, extreme high changes, but definitely there is some evidence for that.
Thank you, Anastasia. I think that's it.
Thank you.
For the moment.
Exactly. As always, if there are more questions upcoming, feel free to put them in the Q&A. And Anastasia, you can have a look at the Q&A section as well, and then see if there's a question that you would like to answer as well. And you can do so by typing. Okay, let's move on. So Anastasia spoke about one of the two big crops that we will talk about today. And the next presentation actually focuses on wheat. So wheat also remains one of the world's most important food crops, but also has some specific challenges like climate change, soil degradation, and growing food demand. So there's a lot of pressure on the wheat production system. And one of the projects addressing these issues is the Wheat Biome projects. And within the Wheat Biome project, we have our next speaker, Rosa Perez Gregorio, and she holds a PhD from the University of Vigo in Spain and studies how food components interact with our immune system, the gut microbiome, and our overall health. So her research uses few techniques to better understand the complex relationship between our food and our health. Rosa, please feel free to take the stage and share your screen.
Okay, thank you for the introduction. I'm going to share my screen. Okay. Can you see it? Yes? Okay. So today I am going to talk about the Wheat Biome project. It's a Horizon Europe project that I coordinate to talk about reconnecting the soil and food. Well, as you know, there are main challenges related to the transformation of agri-food systems, mainly the growth population and the loss of arable land due to climate change. And these are exacerbated because of geopolitical instability. So during the last years, there is a growing field of research focusing on studying the role of microbiota to transform these food systems to be future-proof. And how and why microbiota? Because microbiota play a pivotal role in aerial, aquatic, and terrestrial ecosystems, driving functions such as atmospheric chemistry regulation, nutrient cycling, and also connecting the aerial with the aquatic and terrestrial ecosystems, working in the interactions to maintain the ecosystem's resilience. But if we want to transform our food systems, we need to start looking at the soil, because soil is where the food begins. And in 2023, the FAO published a document stating that to obtain nutritious food, we need to have healthy soils, safeguarding the food safety and food quality, and very especially under these unprecedented times. And why wheat? Because wheat is a strategic relevant cereal in global markets, is the most produced and consumed over the world, playing a pivotal role in global food security, but also in economic endorsement because of rural livelihoods and marginal areas, contributing to sustainable agriculture and adaptability. But the consumption of wheat is also related with several pathologies such as wheat allergy, non-celiac gluten sensitivity, and celiac disease. And in the specific case of these two pathologies, the non-celiac gluten sensitivity and celiac disease, the prevalence of these diseases are being increased exponentially during the last years. And in Europe, the non-celiac gluten-related sensitivity, the prevalence of this disease runs around 5 till 15%, and in Italy, this prevalence is still higher, which is too much. So we need to establish some strategies to control the prevalence of these diseases related with consumption. And Wheat Biome tried to connect soil and food through microbiota in order to understand and unravel the wheat systems to be healthier, more sustainable, and resilient. With two case studies and a lab-scale demonstrator, Wheat Biome aims to understand the biotic and abiotic factors on wheat microbiomes and wheat global quality. And we want to also decipher how indigenous microorganisms have the ability or the potential to be used for designing innovative foods and feeds. Well, in the specific case of soil, we want to understand how biotic and abiotic factors shape the wheat microbiomes and the relationship with these microbiomes with crop quality and resilience. And we have two case studies, as I already mentioned, one in Spain, where agronomic practices, namely the organic farming, conventional farming, and irrigation conditions are being tested. And in the other case study, the soil type and wheat cultivar are being also addressed. With these two case studies, we have the study and the two well-differentiated climatic conditions, the Mediterranean and the oceanic. When we shape the wheat microbiomes and the crop quality and resilience, we want to then identify, validate, and isolate some beneficial microbial approaches to be applied to wheat systems and validate the results obtained in the fields. To finally deliver a decision support systems to help with farmers in a decision maker towards agricultural practices and systems depending on the conditions they have. Well, and what we found regarding the soil and plant microbiota, we have characterized the soil and plant microbiota through 3 different campaigns. And we have considered the pedoclimatic conditions, I mean the physical-chemical properties of soil and the environmental and climatic conditions, but also the agronomic practices and the impact of the genotype. And we have seen that the soil, the microbial communities are primarily shaped by soil type and environmental condition, I mean the pedoclimatic conditions. But the soil-driven structure dominates by resurface and root. Pedoclimatic conditions are the primary determinants of early microbial assembly, and the above ground tissues are strongly influenced by genotype, I mean by the cultivar, and environmental conditions and agronomical practices, so the management can shape these community shifts. We have isolated hundreds of these microbial communities, and we have validated some functionality, mainly with promoting growth bacteria under lab scale demonstrator under draw conditions, and we have a very important and interesting results. Well, to link further the plant microbiota with the nutritional quality, we have analyzed the several traits such as the macronutrients and mineral composition, and we have seen that there is the carbohydrates versus lipids proteins trade-off is dominated the behavior, with fiber affecting in a secondary compartment. And the pedoclimatic conditions and the agronomic practices strongly influence these macronutrients, and the cultivar remains, the significance of the cultivar remains not significant. But when we go deep and analyze not only the total carbohydrates, total lipids, total proteins, and so on, and we characterize the profile specifically on sugars, lipids, and so on, we have verified some differences into the profile. So it seems that actually the microbial communities affects directly the metabolisms of the different cultivar. And regarding the minerals, the mineral profile was mainly influenced by pedoclimatic conditions and agronomical practices, and the cultivar effect is not clear. Regarding the bioactives, the total phenolic compounds, we have followed the effect through these three different campaigns in soil, cultivar, and agronomic practices effect. And taking together the 3 years, we can set that phenolic content was strongly influenced by agronomic management, but also with cultivar. Regarding the gluten content, we have verified that total gliadin is mainly driven by genotype. And with these different traits, we need to correlate now better the different data we have from soil and plant, and in order to understand the whole effect. But then we go to food. We use the grain cultivated under ecological conditions to obtain specific microorganisms to characterize the probiotic effect and the fermentation capabilities to design novel food. We have isolated 45 microorganisms with probiotic properties and fermentation capabilities, and we designed 10 yogurt-like products and 13 fermented, non-alcoholic fermented drinks. And regarding the non-alcoholic fermented drinks, we have observed several impact on nutritional quality of the final food. And this nutritional quality was affected by a pre-treatment, an enzymatic pre-treatment we have made in the wheat flour. And if we treat the wheat flour with amylase and glucoamylase, we have observed that a positive correlation with vitamin B and some minerals. And an extended enzymatic pretreatment leads to release of total dietary fiber and total sugars correlated with the release of short-chain fatty acids and other fatty acids. And the amino acid profile remain in a secondary compartment, not directly related with this pretreatment. Regarding the mineral content, we have observed also a structural degradation during fermentation, linking fiber release with mineral bioavailability. And as Christophe mentioned before, is quite important. Going deeper on the impact on gluten, on food nutrients quality, we studied the impact on gluten proteins profile, and this is a result of the Western blotting analysis, and this is the control of a specific pretreatment with enzymes, and these are 3 batches of fermented food with different consortia or isolated bacteria, and this is the control with another enzymatic treatment and the fermentation. And here is a gliadin control. And as you can see, there is an increase of the recognition of the antibody by proteins. So it seems that the fermentation can increase the immunogenicity of the food. But here you can also see that it's an extension of the hydrolysis of gluten proteins, and the hydrolysis of gluten proteins are directly linked with the celiac disease and non-celiac gluten sensitivity, because gluten proteins are difficult to be degraded by gastrointestinal enzymes. So larger peptides remain longer periods of time in intestine and promote inflammation. But we need to know if this hydrolysis of these or this extended reactivity towards the antibody recognition can have an effect on the human body. So we mimic the human digestion, and we have analyzed the non-accessible fraction. And here in the Western blotting, you can see that the non-fermented food, I mean the control, remain the gliadin intact, but the fermentation improves significantly the digestibility of the gliadin, disappearing completely of the non-bioaccessible fraction. Going deeper and analyzing the bioaccessibility of immunogenic peptides, we can see that in most cases, the fermentation diminished the amount of immunogenic peptides, but 6 epitopes remain persistent to fermentation. So we need to continue exploring different consortia, different isolated microorganisms in order to go further to obtain even healthier foods. We need to link all together and consider the wheat and all of beyond, not only the wheat, but all crops, because there are quite relevant interactions between soil, crop performance, plant microbiota, plant metabolism, and human metabolism, and the impact on human health. To apply a One Health approach, integrating all this data. Thank you for your attention and for this opportunity.
Thank you very much, Rosa. That was very interesting. I can't but help myself to ask that question. When you did the fermentation of the yogurt and the fermented drinks, how was the taste? Was there any specific difference?
The taste is so good. It's a little bit acid, but it's very similar to an Actimel.
Okay.
Yeah. And the beverage is still better because the yogurt, the texture of the yogurt is a little bit oil, is fatty, but the smell and the taste of beverages are quite good. Indeed, we have some in vitro studies of the oral cavity cells the selection of the beverages for being tested then in humans, because we have also a clinical trial in our hands, is based on the results of the taste properties by using this in vitro study, and also the sensory assays and the nutritional quality. And the sensory results are quite interesting. Yeah.
It's great to hear. Yeah, always interested about the taste part of these the products basically that are developed. Looking at Deborah, do we have a question? Maybe one question?
We have one question that are actually two. Will you test the non-alcoholic beverages with consumers? And the second one, existing producers who make similar products?
Yes, actually, to my knowledge, there are not these products on the market. If you are, I mean, I imagine you are focusing on the food part, right? There are not, or at least there are not with the microbial consortia we have tasted, because these strains are specifically found in our wheat and they are not described before. So, and the point is that these are considered novel foods, so we cannot continue with this specific food for human tests. But we have the information to be sent to the EFSA to be approved in case there are any commercial interest on performing this.
Okay, thanks. We have another question. Do we have time, Stefan? Otherwise, Rosa, you can answer in the Q&A box.
Yeah, maybe you can answer that question via text, Rosa, and then we can move on to the final presentation. Okay, great. So far we explored how microbiomes can support crop production, sustainable agriculture, and in the last presentation, we now look into another important challenge for sustainable food systems, which is reducing food waste and creating value from underutilized resources. And technically, we're also going back to the first topic from today, which was food fermentation. So it's kind of like a full circle moment. And yeah, as you may know, the topic of food waste is really a true issue of our time. Around the world, huge amounts of food and agricultural side streams are lost and discarded, but there are some opportunities through which microbial processes can offer some opportunities to transform these materials into useful ingredients and products. And to discuss that topic, I'm happy to welcome Abelardo Margolles Barros. He is currently working as a research professor at the Dairy Research Institute of Asturias, which is affiliated with the Spanish National Research Council. And throughout his career, he contributed to the understanding of intestinal microorganisms and those associated with our food. So, Abelardo, please, the mic is yours.
Thank you, Stefan. I'll try to share my presentation. Let me see. Can you see the presentation?
Yes.
Yes.
Yes. Okay. So thanks a lot for inviting me to this event of the World Microbiome Day. As already mentioned, I am working in CSIC. CSIC is by far the largest public research institution in Spain, and I am participating in the Domino project that was already mentioned. You know that Domino is the acronym of the project harnessing the microbial potential of fermented foods for healthy and sustainable food systems. And I am leading Work Package 4 in the Domino project together with Luca Coccolin from the University of Torino, where we are actually addressing several case studies of fermented foods that I will explain shortly. First of all, when we talk about microbiomes in the food system, it is very important to consider what we call the circularity of the microbiomes, okay? So this means that all the microbiomes in the agri-food chains are connected. For instance, the soil microbiome influences the plants that feed the animals. This in turn affects the state of the animal, and both plants and animal-derived foods influence our microbiota and our health status. But also, for instance, in the farms, the microbes come back to the soil and to the plants through manure effluents. So you see that the microbiomes in the food chains are completely interconnected. You can see it very well in this slide, which is an infographic of the Microbiome Support Association, which is an European association supporting microbiome science and microbiome knowledge that you can check. You can check in this link if you want to know more about this specific association. This figure, I think, was already partially explained by Christophe in his presentation. When we are talking about fermentation, we talk specifically about a raw organic substrate that through microbial metabolism is converted in a different food with different characteristics, okay, when we talk about food fermentation. And normally this food fermentation has a healthy impact. It could improve nutritional characteristics or modulate the microbiota, or it can generate different bioactive compounds, but also it can have a positive environmental impact, okay? It can reduce pollution and carbon emissions, for instance, and it can reduce food waste. Fermented foods have been defined by the ISAP some years ago and by a consensus panel through a paper that was published in one of the Nature journals as foods made through desired microbial growth and enzymatic conversion of food components. And these are some of the values of fermented foods, okay? Might be a source of live microorganisms, improve taste, texture and digestibility, increase concentration of vitamins and other bioactives, remove or reduce toxic substances, inhibit pathogen and food spoilage microorganisms, and benefit human health by reducing risk to some acute and chronic diseases. And then you have mainly two types of fermented foods containing microbes, live microbes, cheese, yogurt, kefir, etc. Okay, you have also fermented foods non-containing live microbes because they have been killing through the processing of the food by heat or by filtration, etc. You can include here bread, heat-treated fermented vegetables, for instance, or wine and most of beers. And then you have non-fermented foods, but often considered fermented foods, okay? That should be made clear, in which no live microbes are used in the production. Cottage cheese, for instance, or fresh sausages. Those are not considered fermented foods. And here in the right part of the slide, you can see a very nice infographic that was actually produced in the Domino project, and in which you have quite a lot of information about what fermented foods are. So let's move to Domino to study novel and traditional fermented foods. As you know, one of the European projects currently ongoing with a focus on the study of fermented foods is Domino, with the overall aim of attributing health benefits to traditional fermented foods and also developing novel plant-based fermented foods which address the changing societal demands. Domino is a large consortium coordinated by Stefan Chailloux in INRA, 10 countries, 19 partners, 12 million euros, and 5 years of projects. And at the end of the project, we hope we can offer novel plant-based fermented foods, some of them tested in vivo in animal models. Very briefly, this is a general overview of the Domino concept with 3 pillars. The first one is the use of synthetic ecology approaches for rationally designed, tailor-made microbial solutions. The second one is to improve the sustainability of fermented foods. And for doing that, we have established living labs around 6 case studies that takes into account the society acceptance of our food prototypes, the food prototypes that we are developing in Domino. And the third pillar is to propose healthy diet recommendations by creating new scientific evidence considering combinations of some health models, mainly human models, animal models, and ex vivo models. And this is the plan of our Work Package 4 in Domino. In Work Package 4, we are designing microbial solutions for healthy and sustainable fermented foods. I know there is a lot of information in this slide, but I will focus only in the main steps, okay? The first thing we want to do is, based on the microbial collection of the different partners and genomic and metagenomic data, we want to design microbial consortia with potential to ferment our fermented foods. Then different factorial screening models will be or have been applied already to test the different consortia in the food prototypes developed in Domino, and the fermentations are being deeply characterized using several omics techniques. This will help us to narrow the number of consortia to be tested, and we will also develop metabolic interaction models to design new fermentations with a lower number of consortia. And at the end of the project, we will select only one consortium per case study and scale up the production of our food prototypes to produce enough amount to be tested in ex vivo and in vivo models. This is basically very, very summarized what we are doing. And here I show you the 6 case studies that we are studying in Domino. In the University of Torino, they are studying fermented olives, traditional fermented olives. In TFTAC in Estonia, they are busy with some fermented vegetables, mainly sauerkraut and kimchi. IRD in France is studying fermented cereals. Chagas is developing novel water kefir products. INRA is focused on the fermentation of legumes. And in CSIC in Spain, we are studying fermented food waste, specifically fermented apple pomace. So in the next slides, I will be focused on showing some information about the potential of food waste fermentation and different possibilities that can be explored. But first of all, I wanted to show you some big numbers about food waste and food byproducts. So every year, 1.3 billion tons with B, this is 1/3 of the food produced for human consumption, is lost or wasted. The percentage varies depending on the kind of food, of course, but for instance, in some of these categories that you are seeing here in the slide, such as fruits and vegetables, the percentage of food losses and waste is very high, around 45%. So these numbers really makes you think what we are doing with our food, and perhaps we have to think that we don't need to produce more foods. We just have to waste less, okay? So you see that this is a huge problem, of course, but this is also leaves some potential opportunities and biotechnological opportunities to valorize this waste. Very briefly, these are publications published in the last years related with food waste and valorization, food waste and fermentation, food waste and valorization and fermentation. I'm not going deeply into numbers, okay? But you really see that 20 years ago, 0 publications. But in the last 10 years, there was a huge increase in this specific topic. So that, of course, points to a growing interest in this kind of studies. So in our lab in CSIC in Spain, some years ago, we started to work with apple pomace. That is the main byproduct of cider making. I'll give you just one example. Every year, we produce a lot of cider in the north of Spain, in Asturias. That is the material resulting for pressing the apple, but it has a very, very low economic value. Normally, it's used to feed livestock or simply discarded. But only in Asturias, there are more than 10,000 tons produced yearly. If we look at the world, millions of tons worldwide, and apple pomace is very, very rich in nutrients and bioactive compounds. Notably, it has a high content of pectin, for instance, that has prebiotic potential. So we are trying to find different ways to valorize this byproduct. One of these ways is the one that we are using in Domino, in the frame of the Domino project. In this case, we design different consortia that include yeast, lactic acid bacteria, and acetic acid bacteria that can ferment apple pomace. And currently, we are characterizing the compounds resulting from those fermentations. But also, we can, for instance, work with pectin. We can extract pectin from apple pomace using different biotechnological processes. Pectin has this structure that you have in the slide. It has a backbone of galacturonic acid decorated with different sugar moieties, and it has a quite interesting prebiotic potential. Actually, we saw that pectin extracting from apple pomace indeed can modulate human gut microbiota, okay? And even we can modify the prebiotic characteristics of apple pectin by using microbial enzyme. In this case, we use carbohydrate esterases from gut microbes. But also other examples can include non-plant-based foods. We can also valorize waste through fermentations from animal-based food, okay? And here you have two examples. Food processing byproducts, for instance. Normally, byproducts are between 30 and 80% of the fish weight, with a lot of potential applications in food, feed, pharma, or cosmetics, or agro-food industry. We know that these byproducts can be fermented by lactic acid bacteria, yeast, and molds, for instance. And for example, fermentative processes using these byproducts supplemented with vegetables and fruits are used to convert fish waste into high-protein supplements for aquaculture feed formulations. Slaughterhouse waste is another example. They are also very rich in nutrients, and about between 30 and 50% of slaughterhouse animal remains is not utilized. So fermentations can be exploited here to convert waste into animal feed, biofuels, or high-value biochemicals. For example, lactic acid bacteria fermented slaughterhouse waste can be used to produce stable and cost-effective pet foods. Another example is biorefineries, for instance. Normally, compared to thermochemical methods, biological valorization of food waste is much more eco-friendly. So this is an example of a sustainable closed-loop biorefinery concept in which the starting material is food waste, and you can use the glycerol that is produced during the obtention of biodiesel to feed different kind of bioreactors and to produce a large variety of bioactive compounds. For nutraceutical purposes, you can produce also ethanol, pigments, enzyme, nanocellulose, etc., etc. And just to finish my presentation, I wanted to give you a couple of success stories, okay? So indeed, you can ferment food waste and you can put your product in the market because we have already some examples. The one on the left is linked to a French company, Green Spot Technology, that is actually one of the companies participating in Domino, that is using vegetable waste to produce fermented flour that can be used for a variety of purposes. For instance, in recipes at homes, in bakery, etc., etc. And another one is this fermented drink on the right part of the slide. You know that dairy whey is produced in very large amount during cheese making. If you use 10 liters of milk, you get 1 kilogram of cheese and 9 liters of whey. So what they did in Arla Foods is a Danish company, is to ferment this whey to produce fermented protein drinks with a variety of different flavors that probably can be quite successful in some population groups, such as people playing sports, for instance, or people maintaining healthy habits. And that's it. That's the end of my presentation. So if you have any question, now is the time.
Thank you very much, Abelardo. That was great to hear. And yeah, I mean, just thinking about the sheer amount of food waste that's happening, like you said, 1/3 of the food produced is being wasted. It's like throwing away every third bag of groceries that I carry home. That is such a huge amount. Really makes me sad, and great to hear that you're working on some solutions for that, and very nice that you could finish with some of the success stories as well. Now, Deborah, is there one question for Abelardo, or should we move to the panel discussion right away in the interest of time?
I'd like to ask him one that was written, and is, what do you think would be the best way microbiome research could support food processing innovative foods in the near future?
Well, you have many answers for that question, okay? Of course, it is very important that funding agencies promote public-private collaboration. That's very, very important. Unfortunately, many research projects do not translate into the productive sector due to a lack of funding instruments, and because the huge bureaucracy associated with national and European costs, that really discourages the participation of some small and medium-sized enterprises in some calls. Okay, large companies, they don't have that problem because they have innovation and research departments, but SMEs need to collaborate with academia. That's for sure for these kind of things. And so funding agencies must be do everything possible to facilitate these private public collaborations.
Okay, thank you. So I would move to the discussion part, and so I invite everyone who has questions to raise a hand and do the question. Otherwise, I will start with one question for all of you, a general question. How would you describe, for your knowledge, people awareness of microbiomes in general? I don't know who wants to start.
I can start with one day. I think it's improving a lot. Okay, the knowledge about microbiome, because these terms microbiome, microbiota actually are relatively recent, from 25 years ago. Previously, we were probably saying flora or microflora, but especially in recent years, people awareness has increased significantly from my point of view. That also because there is a huge information available in the media. Okay, you now have advertisement in TV talking about microbiomes, quite a lot of news in newspapers, but also because the microbiome has gone from being something largely unknown to medical doctors specifically, to something that they truly take into account. This is, for instance, clearly reflected in medical conferences in which some years ago microbiome was not present. Now there is very, very often in the medical meetings, you have specific sessions to talk about microbiome. So anything that can affect our health ultimately captures our interest, that's for sure, which is why people are becoming, from my point of view, increasingly aware of the importance of the microbiomes.
I have another question. Yeah, go ahead.
No, I would.
Rosa.
Yeah, I would just say that I generally agree with everything said, especially with younger people, in terms of their own personal health and microbiomes in general. Like, it is not only a topic that they use for their own health, but I feel like it's also a general, casual conversational topic being brought up recently. So I think that's a very good aspect because, previously we had the good and the bad bacteria, and now people know more. So I think that's a movement in a very good direction.
Rosa, you were talking.
Yeah, because I totally agree. I think that within the last decade, there was the awareness, the consumers' awareness on microbiota was huge, because I think that COVID has a little bit effect on that, because of the bad part of the microbial communities, but also the impact. I mean, the impact can be completely negative, but also positive. And within that, I think that we need to continue educating population about the potential to use these microbial communities for beneficial issues, but maintaining also the alert to pathogens and so on. Because not only on soil and food production, but in our daily routine, our habits are going to affect directly the microbiome that habits our environments. The chemical products we use in the shower, the fertilizers we use in our fields, and also the antibiotics we take sometimes when we don't need to. And I think that consumers are changing, and the vision of microbiota are also changing, also because of the efforts of scientists.
Thank you all. Wondering if we are focusing too much on gut microbiome and maybe overlooking soil and plant microbiomes.
If I may,
Sure.
I think that the information we have of gut microbiome is quite relevant and is useful for soil. I mean, the behavior is such similar in a lot of in a huge context, of course. I mean, if you have a healthy microbiota, you are healthy. If soil has healthy microbiota, soil is healthy. So the problem is that we can use the information of gut microbiota to be applied on soil and plants, but on humans we need to also go across other microbiota, such as oral cavity microbiota, skin microbiota, and the effects of the exposure of environmental factors to affecting directly these microbial communities, and the interaction between these communities and the impact on the health, but also on the environment homeostasis.
I would also agree completely with this, especially because a lot of the methodology is transferable from a field to our literal fields. So I know that quite a few times I've read something about, for example, about low biomass samples and so on and so forth, which was developed for medicine studies, but I used it in soil or plants. So overall, no such thing as too much microbiome studies. No.
Yeah, actually, I fully agree with both. Okay, so it is true that we have been perhaps too focused on human. I don't know if too focused or not, but we have been focused a lot on human microbiome. But instead of seeing it as a problem, we can also view it as something that will help us make significant progress in the study of the microbiomes in the agro-food systems. Okay, the technologies can be translated. You mentioned it already. Although they have been developed primarily for studying the human microbiome, but now we can leverage it and we can use it for our purposes to study the microbiomes in food, in soil, in plants, etc.
Thank you all. And we are basically talking about one health approach, considering all the different aspects. What do you think are the biggest barriers in its implementation?
Well, funding is always a barrier, okay? That's for sure, but not only this one, okay? For instance, sequencing was very, very expensive 20 years ago. Now it's quite cheap. So if you have a project, the problem is not sequencing. It's not the budget, okay? And also, as I mentioned, all the technology developed to analyze human microbiota took a lot of time, and now we have to develop technologies to analyze other ecosystems. But we don't have to start from scratch. We have to adapt them somehow, okay? In some projects, for instance, we have developed new methodologies to study samples with very, very low microbial load, okay? And we have taken advantage from previous methods that they have been using humans, for instance. This is not easy, and it takes time to adapt all these methods to other ecosystems. I see another barrier that I always mention, that is the training of technicians and researchers in these novel methodologies, okay? I don't know what's the experience of the other co-panelists, but these disciplines are quite new, and it is not easy to find well-trained researchers with the appropriate expertise to work in this area. So training is, from my point of view, essential.
Yes, I fully agree, but I think that some strategies are being made through boot camps and summer schools, specifically on omics technologies, machine learning, and so on, because actually we have a problem and we need to overcome training, because this requires a quite specialized area and is difficult, of course. But I think that there are another barrier that is higher, not higher that the budget, because this is the highest, but I think that the point is that there are a lot of different factors affecting the behavior, and it's not you can isolate some factors and study it in an isolated form, that such as in the past. But then when you try to put all together, sometimes it's difficult to have a realistic model to give robust conclusions about the role specifically on microbes on a specific outcome. And I think that the use of IE and the use of machine learning has, or digitalization has give us an advance to correlate all of these different factors, but it's still challenging. And another barrier is the ability or the disability of some microorganisms to grow in environments that are not their own environments. And this difficults a lot to know if the impact of this specific microorganism on the final effect.
I would also like to build up on top of this, because I actually also agree, because there's a large discrepancy between greenhouse lab and field. And commercialization later on. But we also, when it comes to microbiome studies in general, I think there is sometimes present this all-effective view, like this is going to solve a lot of the issues. Well, I think microbiomes are just a part of it. There's so many other factors, like we do need to tackle climate change. We can talk about drought microbiome, but we also need to literally fix what is causing all of these issues. So One Health in terms of microbiome can only go so far, and it is absolutely connected and absolutely influential, but there's larger things at stake as well in there.
Thank you. All good inputs. I'd like now to move to an agriculture perspective and ask you, what is the link between soil microbiomes, crop quality, and human nutrition?
I can answer this maybe, or I mean, all of us can, I think. In general, first of all, there is in terms of soil health, as I've said before, and crop quality, there's an absolute correlation between human health and soil health. For example, there were a few papers that analyzed, for example, soil bacterial diversity and correlation in, for example, are the parasites present in the soil, or do they like appear in the areas around in human health, and so on and so forth. So this is still an ongoing correlation being done. But I think we need to look at maybe a larger scale. With a healthier soil, we have more long-term production of generally better food, which does indirectly influence human health, as well as lesser use of fertilizers, artificial fertilizers, and so on and so forth. So I think that's kind of like a main connection between those things. And I think what's also important to note, when people favor things like organic farming or sustainable farming, very often in stores you would see food with larger prices, and people would buy them because it's better for the health. And ultimately, it is. But oftentimes, when you are buying locally produced organic products, you're also supporting healthier crop production in the region as well. So that's why sometimes you're not just buying expensive product for you, but you're also buying for everything around you. So I think that's also good for people to know.
Thanks, Anastasia. Maria Rosa, Belardo, do you want to add something?
There is such work in the future to continue exploring this, because link soil with human health is challenging research. Because we have some evidences that there is a direct link, but there are only evidences. There are not robust results on this. But of course, as Anastasia mentioned, if you consume local products that are cultivated under conditions that are appropriated for this crop, I think that you are doing the best, because other factors are out of our control, such as climate change, such as contamination. I mean, as a consumer, not as a community. And this is quite important. But I would like to also remark that sometimes the ecologic is not the more nutritious. Sometimes the amount of protein, carbohydrates, and so on is even higher under conventional farming systems. The point is to catch an equilibrium in the farming systems, wherever is conventional or not, okay, and use fertilizers that are going to do not damage our systems, to be sustainable on time. And this is the main point.
Thank you. You mentioned that we have evidence those results. What would it take to get to the robust results? And I'm afraid, I'm worried to hear that it's found.
And yeah, fun, because we need to have additional data, because I mean, if climate change are changing, it's not enough to have 2-3 year analysis under the same crop condition. You need more data, because microbial community is not the major ones, but the minor communities that may play an important role. Why not? I mean, and we need, these minor communities are changing. Not the whole microbiome, but yes, a specific minor microorganisms. And we need time, and we need to have additional data to test our models. I mean, I think that within these years, we are performing models, and then we need to validate how these factors are maintaining or not, and improve a little bit these models by using digitalization, machine learning, and so on.
Thank you. Abelardo, you want to add something?
But I'm not really an expert in this field, but I am sure that there is a huge challenge to correlate soil quality and soil microbiome with human health, for sure. I'm sure that you will have an influence of the soil microbiome in the final product that we eat. But there are many, many links in between, okay? So to really correlate that from the beginning to our health, that's a huge challenge. And it's very important what Rosa mentioned, that we are talking about crops, and we need several years to validate probably our models, and that makes absolutely necessary to have long-term funding, okay? And with national funding, European funding, normally 3, 4, 5 years, that's not enough. So we need long-term plans to validate these things.
Yes, I agree. Another question on this aspect, the agriculture perspective, or regenerative farming measurably influence the microbiome and therefore human health?
I'm sorry, could you repeat the question?
Yes. Does organic or regenerative farming measurably influence the microbiome and health?
So I mean, affection and microbiome can be defined in many different ways. Like as we've mentioned before, like higher diversity doesn't necessarily mean automatically better, and so on. So far, organic farming has been proven to be very beneficial in many ways. But then again, same as the last question, can we correlate it directly to human health in this way? For now, we need more data. In general, is it better for the soil in terms of health and lower fertilizers and pesticides? Yes, in this regard, absolutely. But this is also case-to-case dependent. The definition of organic farming also greatly changes from country to country. Organic farming in the European Union is not going to be the same as organic farming in USA or even within European Union countries as well. So there's a lot to still standardize in a way.
Yeah, in our specific case, through Wheatbiome, we have seen differences into the microbial communities, but not in the functionality. I mean, the ability to transfer the organic matter and the fixing nitrogen and so on is quite similar as a consortium. So this is not actually a problem. And regarding those that Anastasia refer the impact of the region or country in our project, this was quite significant. And I think that the climate has a very significant role on this. I mean, when we analyze the pesticides in conventional agriculture in Spain and in the Netherlands, the difference was completely huge. I mean, we are able to characterize or to identify, in the soil approximately 45 different pesticides in the Netherlands and only one in Spain. But they were applied also different pesticides and phytosanitaries there. And this can be because of not only because of the microbial communities, but because of the high temperature here in Spain, helping to degrade these residues in soil. And this also have an impact on the microbiota. Microbiota have the impact of the pesticides or other contaminants, and it's like a cycle. And it's because of that we need to continue exploring the differences between the countries, between cultivars, between climate conditions, and so on, in order to warrant how we can talk about regenerative agriculture in conventional or in organic, in both, because we need to work for having healthy soils anyway.
I want to add a short question here, is that we need to define exactly what we're looking at. So organic farming is defined by many different things, including like the use of pesticides. But also very often we talk about low tillage systems, which are very influential on the microbiome formulation. So very often when we look at these traits, it's very good to separate them into several categories, which is like the influence of tillage. We have like long-term tillage experience that are very good for microbiome studies. And then the pesticides and like many other things also that could be, categorized.
Yeah, and correlated with the specific factors on this specific region, with all factors in a One Health approach.
With good funding.
That's the base.
I have, Deborah, it's not an answer, it's more a question to my co-panelists, because as I told you, I am not an agricultural expert, okay? But would it be possible to design or to find a microbial fingerprint associated with organic agriculture, for instance, or something like that? I mean, if you are using some specific pesticides and you have a group of microbes that are very, very sensitive to those pesticides, would it be possible to have a kind of fingerprint, microbial fingerprint, that can differentiate organic from conventional agriculture? That would be, for instance, very interesting for traceability, authenticity, or the quality of the products.
There are a lot of cross contaminations, Abelardo. This is quite difficult, and because the contaminants go to the soil and some persist from years. I mean, we have identified some pesticides that are not being regulated in the European Union for decades, but persist there in soil. So it's not easy to do this like a marker, but of course we need to continue exploring this because it could be a nice approach, of course.
I think maybe what I could bring, so maybe we look like for key taxa in organic farming. I'm not sure that that's going to be easy to find or even possible. But what I do know was found, for example, they did, and I have a colleague that does a meta-analysis of the microbiome of, I think it's crops under drought stress, and they have found a taxa there that's kind of enriched under drought stress, for example. So I think that's more possible. And specifically for organic, if it's in a specific region with the same legal requirements for organic farming within the same climate zone, maybe. But anything larger than that, I'm not sure. Yeah.
Thank you all for this discussion. In the meantime, we have 3 questions in the Q&A panel. So what do you think about the secondary effects of incorporating a non-native microbial taxa, biostimulation versus bioaugmentation?
Generally, non-native microbial taxa is almost all of the inoculations that we have. The Bradyrhizobium themselves is not native to Europe nor Brazil, but it is widely used. So this form of addition is quite good. It goes through a lot of checks, as I've written in one of the previous questions, before we even use it anywhere. And also, I think it's important there is things like entire microbiome transfers from one soil to another. For example, that could be maybe what you meant, but that's very early on. And again, combining two microbial communities is always challenging. So we'll see. I think it's a good topic to research.
Okay, another question is from region to region and species to species. How can we synchronize this to improve food production?
Well, we need to first define the similarities between regions in order to give personalized approaches. But as I mentioned, we need to continue exploring the use of microorganisms as biofertilizers, or to explore the native changes on the regions.
Thank you. One last question. The agriculture perspective really sparks a lot of interest. What is the final goal of microbiome research in agriculture? To disentangle individual taxa function in crop performance, a microbial network approach?
Both. But it's impossible to have one only isolated microorganism in the field. So we need to see as a network, but sometimes adding one isolated microorganism can modulate the function of the whole consortia. So both.
Okay, thank you. There is another question, but I'd like to move to the product design perspective. And so if Anastasia or Rosa, you want to answer in the chat. And I'd like to ask Abelardo, do precision and biomass fermentation risk losing beneficial microbial complexity present in traditional foods?
Well, precision fermentation is still a concept that is not entirely clear to me, okay? If we ask a molecular biologist, they would probably say that it involves genetically modified microorganisms designed to produce a specific compounds with high accuracy, for instance. However, from a broader perspective, it can also involve non-modified microorganisms or selected microbial communities used for producing proteins, enzymes, or different flavors. Obviously, in such a highly controlled process, the typical diversity of traditional fermented foods is lost or reduced, okay? You are using one or two or a few microorganisms. However, I do not see this as non-compatible because normally both approaches, traditional fermentation and precision fermentation, can coexist without one affecting the other, I think, because they normally have different purposes, okay? Precision fermentation normally goes to produce very specific compounds, for instance, and traditional fermentation, it can go to ferment biomass, ferment foods, etc., etc. So I don't see an overlap between the two types of fermentation.
If I may, I think that they can combine. I mean, if you put a specific substrate with a specific microbiota isolated or in a consortium, you are going to be able to modulate the expression of certain genes, able to release specific components. So this is such a specific mix of the traditional with the precision fermentation, and is quite used nowadays.
Actually, I think genomics, metagenomics, metabolic modeling, etc., all these kind of things, they help a lot to drive traditional fermentations to specific purposes, okay? It helps you to select the right bacteria, the right consortia, etc., etc., etc. If you have a specific purpose about flavors, aroma, or, I don't know, prebiotic activities, genomics, metagenomics, metabolomics, and all these omics techniques, they help you a lot to decide.
I see. Now that we are heading toward the end, I'd like to ask you which solutions show the most promise for food system and health?
Well,
I would.
So go ahead, Anastasia.
I mean, I don't think there's I think there's multiple of them, but I would say maybe microbiome-informed breeding in terms of plant production, as well as multiple year field consortia trials. I think those are the two perhaps that I look most forward to.
Yeah, I mean, this could be the topic of a different session, I think, because it's very, very broad, okay? There are actually many. If we talk about agriculture, for instance, biofertilizer, biopesticides, or microbes involved in soil microbiome restoration, I think those are quite hot topics now. In animal farming, for instance, health and productivity through the use of microbiomes reduce of methane, for instance, is also a hot topic. In food fermentation, we can use microbiome knowledge to define precisely the characteristics of the fermented foods that we want. We can do it now. 10 years ago, we couldn't. And those are just a few examples, okay? And in humans, for instance, a hot topic now is microbiome-based therapies. You know that in US, for instance, there are some life biotherapeutics, they have already been approved for human therapy, but that's something that it will grow in the future as well, for sure.
Thank you all. It has been really interesting and enlightening, Stefan, to come back live and join me for the closure.
Yeah, thank you very much. Thanks to all of you. Actually, I have a very good idea. I would like to ask you one final question, very, very spontaneous, and I'll explain you why in just a minute. But I would like to ask you, what would be one recommendation that you would give an early career person working on the microbiome?
Don't focus only on the microbiome. Plant physiology, ecology, and microbiome together, if it's the very beginning of the career.
All right.
Yeah.
I would say try to combine computational biology with traditional microbiology, because now we have a lot of experts in computational biology or bioinformatics, but they have never grown a bacteria in the lab, okay? But we also have the other way around. We have wet lab people that they don't know anything about how to build bioinformatic programs, etc., etc. So to combine those two pillars, I think you will have some more possibilities to success in this field.
Yeah.
Okay, and Rosa?
I think that having success and microbial research goes through use multidisciplinary research, as Anastasia referred. Do some work in different labs with different people with experience on that. But the most important thing when you are starting in such a difficult area is good luck, because it's challenging, and also the experts that the microbiologists that they have construct their curricular path for decades on that, they still know nothing about these tiny organisms. There are a lot of information to be explored. So this is exciting. And I think that you need to focus on that. You need to try to learn a lot, but don't be frustrated if you don't achieve all the knowledge you are initially think that you are going to have, because it's complex. And so good luck.
Well, thank you. I think these are all really, really great tips. And the
Wasn't
reason why I
prepared
asked
at
that,
all.
not prepared at all. And the reason why I asked that was actually purely egoistic, because it gives me the perfect opportunity to talk about another event that we are preparing now, which is within the Sustainable Food Systems Network. So within this network, Deborah and I, we're also the community managers of the microbiome community. And one event that we're currently organizing is more directed towards early career professionals, for them to give them an opportunity to share their work, share their resource, and share their ideas most often, and also to connect with other people. And so you see the slide on the screen as well, and I will also post the link in the chat in just a minute. Actually, in just a second, because I can do that right away. There you go. And we would just love to have you there as well. This was just my final transition from this webinar. And besides that, I all I can do is just to thank again to all the amazing speakers that we had today. Christoph is unfortunately not here anymore, but Anastasia, Rosa, Abelardo, thank you so much. It was great to have you and to listen to your projects and your ideas. Thanks also to the attendees with the amazing questions that you provide. It was great to have you engaged, even though the heat was, at least here in Europe, the heat is quite strong at the moment. So yeah, I hope that you have a good time, have a nice afternoon, and all the best, and I hope to see you soon.
Rosa.
Thank you. Oh, I'm sorry. It was a mistake. Sorry.
Thank you all
bye.
again.
Bye.
Bye.
Bye.